The last presentation of this section this morning is Dr. Feng Lin, who is going to talk about myeloid suppressor cells induced by retinal pigmentation. Epithelial cells inhibit autoreactive T-cell responses that lead to experimental autoimmune uveitis.
Okay, first I'd like to thank this opportunity to present some of our data on RPE cells and its possible use for a possible new treatment for posterior uveitis. So what are mdsc, myeloid-derived suppressor cells? Those mdscs, they were originally identified in tumors. And they suppress host T-cell responses, and therefore permitting tumor survival. And in mice, mdsc are defined as CD11b positive and GR1 positive cells. And because of their T-cell inhibitory activity, growing evidence suggests that mdsc have a potential for treatment of autoimmune diseases that involves autoreactive T-cells. However, because it is impractical to isolate syngeneic mdsc from tumors for treatment purposes, new methods of generating large numbers of syngeneic mdsc need to be developed. So as everybody here knows, the eye is an immunoprivileged organ, and there are a lot of ocular cells have been shown to be immunosuppressive. And mdsc are identified in inflammatory eyes. In addition, RPE has been shown to be immunosuppressive. So that raises the question, can RPE induce mdsc? So to test this hypothesis, we followed a well-established protocol for dendritic cell propagation. As many immunologists know, if you culture bone marrow cells with GM-CSF or IL-4 in 5 days, they're going to differentiate into CD11c positive dendritic cells. So what we did is we culture bone marrow cells together with those cytokines in the presence of RPE. As shown here, this is the CD11c, the marker for the DC. If you culture bone marrow cell alone with those cytokines, majority of the cells develop into dendritic cells. But in the presence of the RPE, compared to 74%, only 10% of cells are CD11c dendritic cells, indicating that RPE inhibit dendritic cell propagation. So where did those cells go? We also looked at the marker for mdsc. As I indicated before in the previous slide, the mdscs are CD11b and GR1 double positive. So bone marrow cell alone, you only have like 18% of mdsc, but in the presence of RPE, the percentage increased to 60%, indicating that RPE is able to induce mdsc differentiation from the myeloid progenitors. So do those CD11b GR1 double positive cells has any activity? To test that, we use a well-established CFSE-based cell proliferation assay. So CFSE is a fluorescence dye. You can label the cells. When T cells activated and they proliferate, the dye intensity will be diluted, and you can just check the intensity of CFSE by flow cytometry to look at the proliferation of the cells. For example, here, this is undivided cells. This is cells divided once, twice. So we cultured the RPE-induced mdsc with different numbers of activated T cells. As shown here, without mdsc, you see under microscope, you can see those clusters formed by the proliferating T cells. At a ratio of 1 to 5, the mdsc can almost completely inhibit T cell proliferation, as evidenced by there's no clusters be seen under microscope. And this inhibitory activity is dose dependent. As the number of mdsc decrease, you can see increase of those cluster numbers. And these imaging results are consistent with the flow cytometry results as shown here. Without mdsc, those 60% of cells proliferating, with 1 to 5 mdsc inhibition down to 50%. And as the number of the mdsc goes down, the proliferation goes up, indicating that those RPE-induced mdsc inhibit T cell proliferation. Do they also inhibit cytokine production? To test that, we measure interferon gamma concentration in the supernatant and shows the same thing. It can inhibit cytokine production as well. So what's the mechanism by which RP induce MDSC differentiation? To address that issue, we used a transwell system in which the soluble factors can be exchanged freely, but the physical cell-cell contact is not allowed. As shown here, so the upper panel is looking for the DC differentiation, the lower panel is looking for the MDSC differentiation. Bone marrow alone, you have like 70% DC, with RPE down to 12%. When we put RPE in the transwell without cell-cell contact, the number raised to 51%. Same for the MDSC generation, alone 18%, RPE together 58%, but in the transwell it's 33%. So the efficiency of RP-induced MDSC decreased, but was not abolished, suggesting that both cell-cell contact and soluble factors are involved. Because B7H1 has been shown to be critical for the immunosuppressive activity of RPE cells, we next test if B7H1 is one of the cell surface molecules that's important for MDSC differentiation. So to test that, we isolated Don Smith from the co-lab help us to isolate the RPE cells from B7H1 knockouts. And we did an experiment together with Y-type RPE. As shown here, the B7H1 deficient RPE can inhibit DC differentiation and induce MDSC differentiation almost same as Y-type RPE, suggesting that B7H1 is not important, at least in this experimental setting. So the postdoc was kind of disappointed at that time. So I told him that, okay, let's look at the soluble factors. So he measured IL-6, a cytokine that's well known to inhibit DC differentiation and to promote MDSC differentiation. So he measured IL-6 concentration in those cultures, bone marrow alone, RPE alone, and bone marrow plus RPE, and showing that IL-6 expression is dramatically increased in the cold culture. So to test whether IL-6 has anything to do with the RPE-induced MDSC differentiation, he blocked IL-6 using a neutralization antibody and showed that blocking IL-6 could reduce MDSC generated, suggesting that IL-6 is one of the soluble factors that's involved in the RPE-induced MDSC differentiation. So we showed that RPE can induce MDSC, and those MDSC can inhibit T cell proliferation in vitro, where they work in vivo. To test that, we use a model called experimental autoimmune uveitis. It's an animal model for autoimmune posterior uveitis, which is a common source of vision disability in the US. And EAU can be induced by immunizing C57 black mice with IRBP 1-20 peptide. And it is known that EAU, this model, is primarily mediated by Th1 and Th17 responses against the IRBP peptide. So what we did is we immunized mice to induce EAU, and after immunization, we gave those mice the RPE-induced MDSC by IV injection. And in 21 days, we sacrificed mice, and Cassie from the co-lab made the sections for us. We sent the sections to our collaborators at the National Eye Institute, and they scored the histopathological scores. So the higher the score is, the more severe the retinal damage is. As shown here that compared to those control groups, MDSC-treated mice have significantly less severe retinal injury, indicating that RP-induced MDSC could protect mice from retinal injury in EAU. So Padal from downstairs also helped us to did some imaging. So he used SLO to look at those fundus of those mice. This is one of the pictures showing the control mice, and this is treated mice. And those are the representative histology of the treated and untreated mice. You see the typical photoreceptor folding in the mice with EAU. And those are the thymidine incorporation assay showing that treated mice had much less T cell proliferation, and they also treated mice had increased FOXP3 Treg cells in their lymph nodes. And this is just a recall assay showing that the treated mice has depressed Th1 and Th17 responses. So in summary, I hope I convinced you that we found RPE can inhibit DC differentiation from bone marrow cells, and RPE can also, in the same time, induce mdsc differentiation. And the RPE-induced mdsc differentiation through both cell surface molecules and soluble factors. And the B7H1 is not important, and it's in our experimental setting. And the soluble factor IL-6 could be one of the soluble factors. So mdsc are effective in suppressing autoreactive T cell responses that lead to EAU. So in conclusion, I think we can convince you that RPE-induced mdsc could be developed as a new therapy for autoimmune posterior uveitis, and the induction of mdsc could be another mechanism by which RPE control inflammation in the eye. And thank you.